Hello, good morning, everyone. Let's continue from where we left off from last class. We were filling this chart that compares innate and adaptive immunity, and just do a quick recap of what the difference are. Innate immunity refers to the immunity that you are born with the moment you come into this world, right? Those things are encoded within your genome, right? So they are ready to function as soon as you're born, right? And so the response times is going to be immediate. Okay, they are constantly fighting pathogens that have gained their way into your system, but these are not strategies that are designed to fight off a particular pathogens. They are broad spectrum strategy that would work in a general sense. So that's why in terms of specificity, we put non-specific here. And the antigens are detected by the recognition of PAMs, whereas for adaptive immunity, it takes some time to develop because you need to create a tailored strategy to deal with a specific type of pathogens. Right? I keep on saying, if you get infected by chickenpox, you need to create antibodies that are specifically going to fight off the chickenpox. So that takes some time to develop. And the ability to recognize virtually all kinds of different pathogens out there lies in the fact that we use a gene shuffling mechanism to create unique receptor. So I've shown you last class that if you have a gene that has 4 exons, that alone can create 16 variations, right? Depending which exons you join together. Right? You can join exon 1, 2, 3, 4. You can join exon 2, 3, 4, or just 2, just 4, so on and so forth. So there's quite a lot of variability. With 20 different 20 different exons, you can generate that many possibilities. And we definitely have more than 20 genes, exons, and the genes that would be available for gene shuffling. So the end result is the ability to potentially recognize billions of different types of antigens out there. So in terms of the type of cells that are involved here for the innate immunity, we have we talk about phagocytes, right? We talk about phagocytes like neutrophils, like macrophages. Can put it here, e.g. macrophage. It's too crowded. Let me zoom in a little bit. Macrophage, neutrophils. Okay, so those are examples of phagocytes. And there is another kind of cell that is responsible for killing virus-infected cells or tumor cells. You might remember that is called natural killer cells, or NK cells. Okay, so these are the kind of cells that are involved in innate immunity. On the other hand, we have the adaptive immunity, and the type of cells that are involved are T cells. And as we will learn today, there are different types of T cells, as well as B cells. Okay, so in Unit 2, Lecture 2, we talk about how T cells are responsible for coordinating the immune response, telling other cells what to do, and the B cells are responsible for producing antibodies. And there's one last category here, which is called the immune memory. How persistent is it? So for innate immunity, it is no immune memory, none. Okay, you won't remember what you fought off. Like, if you touch a table, dirty table, and then you eat food with the dirty hands, okay, you're gonna have to fight off all the pathogens that you introduce into your mouth. Next week, you do the same thing. You touch the dirty table and then you eat food with your dirty hands. Even though it's the same kind of pathogens that you introduce to your system, you got to fight them off from scratch, okay? So innate immunity does not remember what you fought off previously. Every time you have to start from scratch. Whereas adaptive immunity, they are long-lasting immune memory. If you fought off chickenpox once, you're going to be able to fight it off. You're basically going to have protection for your life, okay? So those are the key differences between the innate and the adaptive immunity. Does anybody have any question? Okay, great. So that is that. Let's move on to our lecture slides here. And we have quite a bit of note to take today. But let's do a few slides first. I got a new mic, by the way, so you shouldn't hear me cutting off again. But if I do, just send me a message, okay? Or unmute would probably be better. Just tell me right away. So, because sometimes I can see the message when I'm going through the slides. All right. So now we're going to talk about adaptive immunity, right? So another name for adaptive immunity is acquired immunity. Okay, acquired immunity. If the innate immune mechanism do not quickly eliminate invading pathogen, if the mechanisms of innate immunity, like the inflammation, the phagocytosis, the complement, if all those things are not clearing the pathogens quick enough, then you might have a serious infection. And by that time, it is important to trigger the adaptive immunity, right? To create a more tailored solution to fight off whatever invading pathogens you have. And so here are some characteristics associated with adaptive immunity. Okay? First of all, it has the ability to distinguish self versus non-self, okay? And that means that you need to be able to tell what belongs to the body and what does not, right? You don't want to end up attacking your own self, right? So that's a key feature of immunity in general. Memory. Memory is the ability to remember an antigen and make faster, stronger response if you encounter it again. Now, here is what people get a little bit confused, because people see antigens on some pages and they see pathogens in other pages. And they're like, what exactly are we fighting off? Are we fighting off the pathogen or are we fighting off the antigen? So just to briefly clarify it, and then we're going to talk about it in the upcoming slides again. Pathogen is the virus, right? Is the bacteria, is the fungus that's invading your body. It's the entire organism, okay? But on the surface of these pathogens, on the surface of the bacteria, on the virus, there are molecules which are the antigens, right? So the immune system recognizes these foreign antigens, and then which in turns helps them recognize the pathogen themselves. They're not going to look at the entire bacteria because that's way too big, right? They're just going to be checking the antigens that are on the surface, and then they want to know, hey, is this antigen that we also have on the human cells, and is this something that you already have in the body? And if it's not, then your immune system recognize that antigen as a foreign antigen, and then you will respond to it accordingly. Okay. And there are two additional features related to adaptive immunity. One is called specificity. The other is called diversity. So the best way to visualize this is to think of a whole set of screwdrivers, okay? Each screwdriver is different at the tip, and the tip allows you to tighten like screws that has different grooves, right? Some are flat, some are cross, some are hexagon and whatnot, right? So they don't really work with the different types of screw. Okay, like you cannot. I don't know what they are called. Like they actually, all of them has different names apparently. So like if you think of a screw, if you have a screw that has like a hexagon in it, like you would not be able to tighten it with a screwdriver that has like a flat end, right? The flat end is only going to be good for tightening the flat head screw, right? And so that's what specificity means. Okay, each one is only good for a particular screw set. So each adaptive immunity response is only designed, is only tailored for one particular pathogen, okay? Chickenpox, for example. And it would not work if you have another infection. It would not work if you get like hepatitis. However, when considered collectively, when you think about like the whole set of screwdrivers, then you can tighten any kind of nuts out there, right? Any kind of screws out there, right? And so when you consider adaptive immunity as a whole, it is very diverse. It has diversity because of the gene shuffling that we talked about. It has potentially billions of different antigen receptors. Okay, so each receptor is designated for one type of antigen, but collectively, right, all these receptors, it gives you the ability to fight off virtually anything. So that's the difference between specificity and diversity. The ultimate weapons that we have against pathogens is basically antibodies. Okay, that's the whole point of the adaptive immunity. It's to create antibodies that would attack the invading pathogen. And the antibodies, as you probably remember, they are created by the B cells, right? Okay, so each antibody is going to be having a Y shape, right? So if you think of the letter Y, right, that's what the antibodies look like. And the trunk of the Y, that's what we call a constant region. Constant region. That's the region that does not change. It is the same for all the antibodies. Okay? That's kind of like the handle of the screwdriver, if you would. It's the same handle for all of them. What difference between the antibodies are the variable region here. Variable region. Okay? Each of them will be slightly different depending on how the gene shuffling was done. Okay, so in this example, you can see the groove here would recognize the antigen that is on this virus. So this whole thing is a virus, right? Virus, and then these little hexagons that are on top, those are the antigens. Okay, remember, we also have antigens. So it's not so much that the antigens are bad, but it's the fact that these are foreign antigens. These are antigens that you do not have in your body. And that's why we're tagging it with the antibody. Okay. Over here, we have another antibody. And you can see the constant region is the same, but over here, the variable region, the groove pattern is different, right? So these would be designed to recognize, those antigens, right? The semicircle antigens that are found on the bacterium. Over here, bacterium. Okay, so one antibody for one antigen. So generally, and generally is the key word here, generally each antibody will only recognize one antigen. Okay, so the one that recognizes the pentagon will not recognize the semicircle, and vice versa. Okay, but there are exceptions that we will talk about in just a bit. Is this okay? Does anyone have any follow-up question? Okay. So what happens after you produce the antibodies? How do they actually work? Well, antibodies can work by several mechanisms, and I'm gonna show you some of these. The antibodies can react with virus and toxins to neutralize them. Right? So here are some toxin molecules. Toxins are basically poisons that are made by bacteria. Bacteria, sometimes the presence of the actual bacteria would be causing harm to your body, but other times it's not so much the bacteria themselves that's causing the harm, but the poison that they produce, right? These toxins that they produce. Example of that would be like tetanus or botulism, right? Even if you kill off the bacteria, the toxins that they have produced can still cause problem in your cells. And so, you know, in the presence of these bacterial toxins, it's going to damage the cell, perhaps kill it off. But if you have antibodies, the antibodies will bind to the toxin and neutralize them. Okay. That is also something that could happen when you get bitten by a snake. Right? You can get serum, which would help neutralize the toxin, the venom. Another example is virus. The virus, as you probably know by now, they will go into cells and then multiply inside the cell. But some of the antibodies will be able to bind to the virus and cause them to not be able to attach to your cells, and therefore no infection, right? And that is basically one way of how antibodies are able to help clear an infection. We call this process neutralization. The second one is something that you are already a little bit familiar with, and that is the clumping action. Remember in the RH, not the RH, but the blood typing, right? Or blood transfusion. If you transfuse the wrong kind of blood, the antibody in the plasma of the recipient will bind to the incoming red cells and create these agglutination, right? These blood clumps, right? So that can happen to pathogens as well, right? So you can have bacteria, and then you know the antibodies is going to bind to it, and then another bacteria, and then you have more antibodies, and ultimately you end up having this large agglutination, large immune complex, right? And that is just much bigger, much easier for the macrophage to eat up compared to if you only have one bacterium. Okay, so like this is really small, and then the macrophage it's still going to eat it, but you know you're just eating one at a time. But when you have all these things clumped together, you are able to pick it up much easier. I don't know if you guys like sweep the floor at home or something, right? But then if you ever sweep the floor or like rake leaves, right, you make it into a big pile, and then it's much easier to clear it than, you know, clearing it one spot at a time, right? So essentially this is what's happening, okay? The antibodies kind of gather all the bacteria into one big lump, and then you can just, you know, either vacuum it up or like, you know, if talking about the leaves, you can just pick up a whole scoop and put it in the bags, right? So it makes the process more efficient, makes it easier for the phagocytes to engulf. Hope that makes sense. And the last thing that antibodies are able to do is that they could help activate complement. Not only does the antibody clump the bacteria together, but the presence of the antibody on the bacteria can actually help activate a complement. And you might remember from last class, one of the functions, one of the three things that we talk about regarding complement is that they are able to form these membrane attack complexes, right? The MAC, right? So that will punch holes in the bacteria and it will kill them off, right? And once they're dead, the macrophage can come and clean up the dead bacteria as well. Okay, so That's why we need to produce the antibodies, and that's like the final weapon your body has against pathogens. Okay, if you don't produce the antibodies fast enough during the period which the adaptive immunity is kind of producing the antibodies, getting ready to produce them, the pathogens can do a lot of harm to your body as well. So by the time you make the antibodies, it might be too late to fight it off, right? Or you might make the antibodies, but the antibodies is not strong enough to clear all the pathogens. They are maybe multiplying faster than you can clear them up, and that's how basically you could potentially die from an infection, and that's why you need like modern medicine, right? You might need antibiotics to keep the bacteria at bay, and then your own immune system will complement the action of the antibiotics and help you heal faster. If you have a viral infection, you might need to have antiviral drugs, right? For the same reason, right? You need to have a two-pronged approach to deal with the infection. But as far as your body goes, antibodies is the final ultimate weapon. Okay, if you don't have medicine, right, antibody is your only way out of the infection. Okay. Now, we are gonna talk about this concept of epitope and antibodies. And to illustrate this, I'm just gonna play a game with you, okay? It's really quick. If you want to unmute and answer, great. If you don't want to talk, you can type, or you can just think about it in your head, okay? It's just gonna take a minute. So I'm gonna describe an animal, and you're gonna tell me what kind of animal I'm talking about, okay? So this animal has a very long nose. What am I talking about?
Anteater?
Oh, interesting. That is the first time I hear people say that. But I was thinking about elephant. Okay, I guess you would be right as well. Okay, so elephant. Good. Okay, next one. This one has very long neck. Okay, what am I talking about? Super long neck.
Yeah.
Giraffe. Exactly, right? Okay. And here is the last one. Okay, this one has a tail. What am I talking about? Well, you're like, that could be anything, right? Because anything has a tail, right? It could be a lion, could be a tiger, could be a cat, could be a dog. I'm not sure what you're talking about, okay? So here's the thing. Some antigens are more common than other antigens, okay? Remember, antigens are molecules that are found on the surface of a cell, right? Okay, so some antigens are unique features to that specific bacteria, for example, okay? Just like the long neck. As soon as I say long neck, right, 9 out of 10 people will immediately think about the giraffe, okay? When I say the long nose or the trunk, right, then I'm talking about elephant. But some antigens are very common. It's not a unique identifier of the bacteria, such as the tail, right? And it's really common, like everybody has that, right? So in terms of coming up with an antibodies, you want to create antibodies that would recognize some of these unique features, okay? So these unique features are what we call the epitopes that are on the antigen, okay? So this is the antigen which is on the bacteria, okay? So this is not to scale, by the way. Okay, like this is much smaller, but then on the antigen, there are like different spots. Some spots are more unique than others. And it turns out the antibodies are more potent. They are more, what's another word for potent? They are stronger. They are stronger, more effective if they are able to recognize something that's unique, okay, rather than something that is commonly shared between many different pathogens, okay? So rather than creating an antibody that recognizes the tail, that's not going to work too well. We want to create an antibody that recognizes the long neck, for example, right, to recognize the long nose. So immediately you will be able to generate protection. Okay, so you can see on the antigen, some parts it's only weakly binding, but then some you have binding for like both of the arms, right? And that one is the one that will be stronger. So when we have a vaccine, for example, which we'll talk more about later, if you're introducing something to generate antibodies in the system, you want to find a part of the antigen. You want to find a part on the antigen that will create these strong antibodies with the double binding. Okay, in other words, you want to find an epitope that is going to create a strong immune response. So that's what it is. Anyone have any question? Pathogens, antigens, epitope, antibody. Great. All right, let's quickly do this poll question here. We'll see how it goes. Oh, by the way, I haven't told you, we haven't learned it yet. I guess I put this question in the wrong spot. But plasma cell is a type of B cells, as you will learn. Okay, so this is correct, okay? You should choose C, but check the other ones. Okay, let's take a look. Fasana, I think this one's I think I understand why you're asking me that. But this one looks for like all correct answer, right? So like C is not the only one. So that is correct, right? They are Y-shaped proteins. Is that why you're asking that, Tashana? I just want to make sure I didn't misunderstood you.
Yeah, because you said C is the correct answer, but then
Oh,
I was
C is
wondering.
one of the correct answers. Yeah, sorry.
Oh, okay. Because I was wondering, I'm like, am I mistaken? Because on the diagrams I
No,
could
no, no.
swear it
Yeah,
looks like a Y.
it is Y. Yeah, you're correct. Okay,
Okay,
so this is like an
perfect.
example of like a multi-select question, right?
Okay, okay.
Yeah.
Thank
Sorry
you.
about that. Antibodies produced against one pathogens will generally work against another. So that's not true, right? Will generally not work against, as I told you. Antibodies are found in the blood plasma. That is also correct. Okay. Variation in antibodies arise from gene shuffling. That is also correct. Okay, so all these things are true except for B. Okay, class note. All right, so we're gonna take a super big note on antibody production. You're probably gonna need some colors, okay? Because the diagram only will make sense if you use color. 4 colors maybe, if possible. 4 colors. I'm just gonna keep on going with the note here from last class. Okay, this is a long note, all right? So brace yourself. This is adaptive immunity. And we're going to talk about antibody production. Okay, so like you should put your paper like this. We're going to start here, and then we're going to have some stuff coming down, And then ultimately we're gonna go this way, and then the note is gonna end over here. Just so you plan ahead, right? To know where we're going for. Okay, so let's backtrack a little bit. Let's say a person is infected by some bacteria. Okay, I just use bacteria as an example because people can easily identify with that. Okay, but really it could be anything. It could be virus, could be fungus, could be parasites, and what have you. All right. So here we have some red bacteria, little red oval. Please use colors. Okay. The note will not make sense if you don't use color. I don't care if you use red or not. Well, preferably you use red. The red actually means something at the end, okay? Everything else you can just use whatever color, but try to stay with the red for the bacterium. And everything else, you can make your own choice. So these are bacteria. Bacteria. Okay. So for those of you who, like me, English is not the first language. Bacteria is plural, and bacterium, I-U-M, that's singular. I didn't know that before. In case you're wondering the same thing. So these are bacteria, and you get infected by it, right? So let's walk through this process. You get the bacteria in your system. What are some of the things that happens immediately? Well, if the bacteria is already in your system, then that means your barriers has failed, right? The physical barrier was not able to keep them out. And the chemical barriers was not able to wash them off. So now they are in your system. And what happens is you're going to have the inflammation. You're going to maybe turn on some complement. You might have a little bit of fever, although fever at this point is probably too early. The fever kind of kicks in later on. But inflammation happens almost within the first hour. And then, the complement is going to work right away. And also, you're going to have the phagocytes, right? So here I'm going to draw a macrophage. A macrophage. So on these bacteria, there are going to be various types of bacterial antigens that we don't have. There are going to be special molecules that would be part of the PAMs, the pathogen-associated molecular patterns, the PAMs that are recognized by the macrophage. So the macrophage is going to come, and then they're going to eat them up. Okay, eat them up. It's going to look like this. Let's just keep it small. Okay, so there we go. And is going to surround the bacterium in a vesicle. Okay, so this process is called phagocytosis, right? So far, nothing new, right? We learned all this already. What I did not tell you is what happens after the bacteria has been engulfed, has been phagocytized, right? Now, we learned about two types of phagocytes, right? We have the macrophage, and then we have the neutrophils. We're going to focus on the macrophage because macrophage has a special ability that the neutrophil does not. Okay, and that special ability is something called antigen processing. Excuse me. So antigen processing is the ability to basically digest the bacteria and display its antigen on the cell surface. It's probably easier to understand if I draw a picture for you. So what that means is the macrophage will eat the bacteria, it will break it down, and then it will take some of those bacterial bits, the bacterial antigens, and put it on its surface. Put it on the surface of the macrophage, that is. Okay, so that's the bacterial antigen. Okay, it's really proud of what it did. It's like, yay, I ate this bacteria. I'm going to show everybody. So it's going to put the display on the surface of the cell membrane. This ability to digest the bacteria and put it on the cell surface, that is called antigen processing. Like I said, this is a very long process, the whole note. So I just want to go slow and want to make sure you understand what I'm talking about. Any question? Min said kind of like spiking the enemy. Yeah. Exactly. If you're into like, Game of Thrones, that kind of stuff, that's exactly what's happening. But try to keep it, rated PG, right? So we have the bacterial antigens on the surface. So any questions about what's happening? Now, none of this is adaptive immunity, right? Everything that's going on right now, it's still part of the innate immunity. All right. Hopefully, okay. Now we're gonna switch gear. We're gonna go a little bit to the right. Remember your note, right? You should be doing this landscape, right? As I said, so you have something here now. Now we're gonna move on to the other corner. All right, and go all the way to the right. Okay. I'm telling you this because I've done this so many times, and then when I do this in classroom, halfway through the notes, I hear students grunting. They're like, "Oh, I'm out of space. I don't know what to do." That's why I'm telling you where to put things. Okay, so on the top right corner of your note, okay, we're gonna have this thing. Hopefully, you know what I'm drawing here. That is bone. Okay, and at the center of the bone. What's so important in the center of the bone that's related to what we're talking about?
Bone marrow?
That's right. That is the bone marrow right here in the center. Bone marrow. And you probably remember in the bone marrow, we have something called hematopoiesis, right? That is the process of creating new blood cells. So from the bone marrow, we are going to have these white cells coming out, and the one that we want to focus on is something called naive T cells. Okay, naive T cells. Now you heard of T cell, and then now it's like naive. What does that mean? A naive T cells, I'm just gonna keep it very simple. A naive T cells is basically an immature version. Okay, immature T cell that has not been activated yet. Okay, they don't know anything. They are naive. That's how I describe my kids. Right? Oh, so naive. You haven't seen the world. You don't know the dangers of the world. Naive T cells, in a similar way, they have not been activated. They haven't met the dangerous pathogens of the body. They cannot fight. They are naive. Okay, immature T cells. Now the T cells will actually migrate to the thymus. Okay, they will continue to mature at the thymus. And then there's something that will happen. Okay, remember they have to pass the tolerance test. We talked about that. And we're just gonna draw a bunch of T cells here. Okay, so that's T cell, E cell, T cell. That's why they're called T cell, by the way. The T stands for thymus, okay? T cell. So you will have a population of these naive T cells. And this is where the color comes in, okay? The naive T cells, they each possess some kind of receptor, okay? So this one might have a blue receptor. I'm just using the color as a way to distinguish them, right? So this one has blue receptors on it. And of course, the receptor is actually proteins. And due to the gene shuffling that we talk about, the receptor will have different shape. Okay, so it's difficult to draw so many different shapes. That's why I'm using color instead of shape to distinguish them. Okay, so this one is going to have a blue receptor, right? This one might have a green receptor on them, and this one might have red receptor. Okay, and then you will have another one like a purple receptor or something. Okay, so I'm just going to put a note here. Each naive T cell. Oh, and again, right, like for those of you who are wondering, the naive, the I is like, I think it's, I don't know, like French or something. That's why there's like 2 dots on it instead of 1, okay? But you can just spell it with 1, doesn't really matter. Each naive T cell has a unique receptor, okay? Created by gene shuffling. Okay, and they are once again represented by different colors in my diagram. Again, just another checkpoint to make sure all of you are okay. All right. Let's see what's going on next. Now back to our original happy macrophage that has just ate the bacteria, processed the bacteria, and put it on its surface. So what this macrophage will do now is something called antigen presentation. That's next. Okay, antigen presentation, right? The macrophage will show will present the bacterial antigen to naive T cell. Okay. So basically, you know, if I have to tell it like a story, the macrophage will go to this naive T cell with the blue receptor. It's like, hey, we are infected by these red bacteria, and that's what they look like, right? Check out the bacterial bits that I have. Will you be able to help fight this off? Well, this naive T cell, because it has the blue receptor, right? It's not suitable in fighting off the red bacteria, right? In other words, like the shape don't match, right? The shape of the receptor does not match the shape of the antigen, right? It's not going to be able to help with the infection, right? So this one is not going to be selected. And then the macrophage will move on to the next one. Hey, we got infected by these red bacteria. Are you able to help? Right. So in this case, right, this cannot help, this cannot help, and this will be the chosen one. Okay, do you understand? Naive T cell with the correct receptor will be selected, will be chosen. And it gets activated. Okay? That's basically what's happening. Remember, you don't only just have 4 naive T cells at any given time, right? You have like a whole pool of them, right? And it's very likely you already have a T cell with the required antigen sorry, with the required receptor to fight off whatever infection you have. But sometimes you don't, just because of bad luck, right? Okay, these are all randomly generated, and you know, maybe you don't have it yet, right? So it takes longer to find it, and then your infection would last longer, because now instead of making the antibody in a week, it might take you like 2 weeks. So you might feel, you know, under the weather for a little bit longer. Is that okay so far?
Professor?
Yeah, go ahead.
These naive T cells, these
The
naive T cells with the receptor, they are not antibodies?
They're not antibodies. Yeah, they are just receptors, like a protein receptor. Okay. So just to elaborate on that point, right? There is a key and lock relationship between the T cell receptor and the bacterial antigen. Okay, so that's another way of thinking about it. Okay, we have a lock here, and then these T cells, they all have a different key. Which key fits? Okay, that's the job of the macrophage. They have to find the one with the right key, right, with the right receptor, and then that one will be the chosen one. Okay. So because the macrophage is able to do antigen presentation, macrophage is sometimes, not sometimes, okay, macrophage are also known as antigen presenting cell, APC, antigen presenting, right, cell. Not all phagocytes, not everything that can eat things is able to present antigen. Okay, so neutrophils, for example, even though they're able to eat up the bacteria, even though they are phagocytes, but they cannot do the presentation. Okay, so those are not APC. What do we need to activate a naive T cell? We need an APC. Okay, we need an APC. So macrophage is an example of APC. There are other APCs in the body, but we're not going to worry about it. Okay, so just let me repeat that. In order to activate a naive T cell, we need to have an antigen presenting cell. And the only example of antigen presenting cell that we've learned is macrophage. It eats it up, puts it on its surface, and then it's going to present it to the naive T cell. And if the T cell has the correct receptor, it will be chosen. So that's the story so far. Let's take a look at what happens when you get chosen. Okay, so this thing is going to get chosen. Just going to go over here. Once chosen, they will undergo clonal expansion. Okay, so this is just basically mitosis to make many copies. Okay, so we're gonna mass produce these things. Yeah, I think that's enough. Okay, and each of them would have the red receptor now. Okay. And afterwards, you're gonna have like a whole pool of these naive T cells, right? Okay. And they will differentiate into different specialization. Okay, so there are 4 different paths that they could go to. Okay. Let's make this longer. Okay, we'll do that one last. Okay. We're gonna go over here. So some of them, they are going to become what's called killer T cell. Okay, so I'm gonna give this cell like a gun or something. Okay, so that would be a killer T cell. That's actually what they're called. Okay, like I'm not making this up. Okay. Another name for killer T cell is a cytotoxic. Okay, it means the same thing. Okay. And the job of the killer T cell or the cytotoxic T cell is to kill infected cells. Okay. So that sounds a lot like the NK cells, right? But it's important to distinguish them. NK cells are broad spectrum, innate response. This one is tailored for the red bacteria. And make no mistakes, bacteria can sometimes enter a cell and infect them as well. So that would be the job of killer T cell, to kill them off. Okay. We're gonna have another kind of possibility. These are called memory T cells. Okay, so I'm gonna put like a little thinking bubble here, right, to show that they remember. Right, so these are the memory T cell. Right, so these will stay in the body for a very long time. Right, to help kickstart immune response if infected by the same pathogen again. All right, so next time you get infected by this red bacteria, fear not, you don't have to go through the whole selection process again, right? You immediately will pull this up and you can start fighting it off much quicker. Okay, it's like creating a file, right? This is a known criminal. We've seen him before. We know how to deal with him. So when they strike again, you will be ready to fight. Okay. And when it's all done, when this is all finished, when you clear the infection, you want to stop the white cells from dividing, right? You want to stop the clonal expansion and all that. And this is where you need another type, right? This guy is going to be holding a stop sign. Okay, and this one is called the suppressor. Suppressor OR, I think suppressor T cell. Right. It will slow and stop the immune response once infection is cleared. Okay. We don't want uncontrolled cell division. Right? If the white cell keeps on dividing in an uncontrolled manner, that's bad news. Right? That's kind of what leukemia is, for example. We definitely don't want that. Okay. Do you guys understand, right? So after the naive T cell has been chosen, they clone themselves and then they transform into these outcomes. Okay, so all these different outcomes, they are called effector cells. Okay, so how do I put it here? I'm just gonna put it in red maybe, right? Differentiate, that's the word. Specialize, right? That's what I mean. Differentiate into effector cells. Okay, so there are 4 paths, right? It could differentiate into suppressor T cell, memory T cell, killer T cell, and then there's this one last one that we're going to talk about in a bit. again, I just want to pause and let you, catch your breath and see if there's anything you want me to clarify. I hope it's pretty straightforward, right? And the story makes sense so far. We haven't made any antibodies yet, by the way. Okay, we're not even there yet. We're on the way to do that, but these are all the things that happens before making the antibodies. All right. So the last type of effectors for T cells are these things called helper T cell. Helper T cell. And I guess I will just draw a hand on them, like a helping hand, right? I don't know. I haven't thought. If you think of a better way to represent them, looks like little wings. If you think of better ways to represent them, please do let me know. These are called helper T cell. All right. So let's go back to our diagram right here. Coming out from the bone marrow, we have the naive T cell, sure. But also we have the naive B cell. Okay, all these blood cells come from the bone marrow, right? Okay, naive B cells. And just like the naive T cell, the naive B cells are going to have a population, okay, and they have unique antibodies, okay, unique receptors that will correspond to the antibodies. So, I'm just going to use the Y to represent them. Maybe you will have one that has the pink antibodies. Maybe you have one that has like, blue antibodies, and some of it's going to be the same as before, right? Green one, red one, and, let's throw in like an orange one. Okay. So each naive B cells would have, again, a unique receptor that is created by gene shuffling. Okay, so I'm not going to rewrite that. You probably, if you understood that, you will understand this, right? And so the helper T cells function is to find and activate naive B cell with the correct antibody. Okay, so like, are you gonna be able to help? Nope, 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 nope. This is it, right? This is the chosen one for the second round. Okay, and once chosen, it's the same story now. Right? This is gonna undergo clonal expansion. And you will have a whole lot of them. Okay. And once you have a population of them, oops, oh boy. Once you have a population of them, oh, come on, pen's not working. Once you have a population of them, they will now be able to differentiate again. Okay, they will be able to differentiate into effector cells, just like before. Okay, so differentiate. Differentiate means to change and to specialize into effector cells. Okay, but there are only two choices. If you're a B cell that gets selected, you only get to become one of two things. Okay, you will become plasma cell, Okay, they're a little bit like rectangular looking usually, and they are basically antibody factories. Okay, they will pump out a bunch of antibodies, and that's what you want, right? That's what you want. You want to have the antibodies, the red ones, that would be specifically unique for fighting the red bacteria. Okay, so these antibodies will neutralize, right? Neutralize toxin, perhaps. It will form immune complex, right? Activate, complement. We talked about these things in the PowerPoints, right? Remember the PowerPoint slides? Right, so that's going to help clear the infection. If you don't aspire to become the plasma cell, the other option is to become a memory B cell. Okay, so same thing. It's gonna have the little memory bubble. Okay, and memory B cell. It's the same same function as the memory T cell. Okay, I am just gonna copy it, and then you can write it down later. Okay, it's the exact same function. Right, so if you have fought off chickenpox before, you will have some memory T cells for the chickenpox and some memory B cells in the chickenpox in your body right now. I know I keep on asking, but seriously, like, does anyone have any question? It's a very long process.
I kind of have a question because, like, sorry, it's a lot because I might have already written it down, but
Yeah,
the difference between the T cells and the B cells, because if the memory B cell does the same as a memory T cell, what's the point in having both of them?
Yeah, that is a great question. Okay, so the memory B cell. So let's say the next time we get infected by the red bacteria, right? Immediately the memory B cell will be able to pump out antibodies. Okay, but you don't have like a huge number of memory cells in your system, because then that's like a waste of space almost, right? So you might have like a few that would be able to immediately pump out the antibodies. So that will keep the infection from getting out of hand. However, just to make sure that you're able to clear it, you also want the memory T cells, which would immediately be able to sort of go backwards and trigger all these things again, so then you would get more helper T cells, and then you will be able to mass produce more B cells. So the infection that you get the second time is usually stronger and much more potent. Okay, people who had secondary COVID infection, sometimes they say, oh, it's like much stronger the second time, right? And sometimes we do a booster shot, right? Okay, and when people get the booster shot, the second shot for the same immunization, right? Like a booster for hepatitis, booster for COVID, right? The side effect could be much stronger the second time, and that's because of this really ramped up response to the same antigen, right? Okay, so not only do you need the memory B cell to immediately make the antibodies, but the memory T cell would kind of ramp up the whole thing, just almost like an overreaction, to make sure that whatever is infecting you the second time is not gonna be able to spread in the body. Does that answer your question?
Yes, it does. Thank you.
Okay, all right. So anyone else? Everybody happy? This happens every time you get infected with something like serious, right? Okay, like influenza, cold, chickenpox. RH mismatch is the same situation, right? Except instead of having bacteria at the beginning, you have these RH positive cells, and your body goes through this whole ordeal to make the anti-RH antibody, right? Okay. So I know this is already very busy, this diagram, but I need to add 2 more things, okay? Just to finish up. All these things. So if you have a highlighter, you can highlight all these things that are going on with the T cells. That has a name. Okay, this is called. I cannot write in yellow because, you know what? Maybe I can. Okay, so all these things, these are called cell-mediated immunity. Okay, because much of the action in terms of like fighting the infection is carried out by cell, right? Killer T cells, right? Helper T cell. So these are cell-mediated immunity. On the other hand, down here, these ones that are done with the by the B cells, these ones, they are called antibody mediated, or some textbook calls it humoral immunity. Okay. So adaptive immunity kind of have 2 main branches, right? We have the T cell department, the cell-mediated immune response, and we have the B cell department, which is the antibody-mediated immunity. Okay. Now, just a final recap in case you know, you got lost in all these process. So basically, we have the naive T cell. And what do we need to activate the naive T cell? What should I put on top of the arrow here? What kind of cell is needed to activate a naive T cell?
Macrophages with the antigen?
Yeah, so APC, right? APC, antigen presenting cell. The one that we learned is the macrophage. And that will cause it to become the 4 things, right? Okay. You know, the helper T, the killer T, the memory T, and the suppressor, right? Suppressor comes later, of course. No point of making the suppressor early on in the infection, right? But ultimately you need them. Okay, so that's that. And what about the naive B? Naive B cell. What do we need to activate a naive B cell?
Helper T cell.
That's right. So we would need the helper T here. Helper T. And then we would get the plasma cell. And memory B cell. Okay. So APC, I'm going to put star here. APC is very important because these serve as link between.
Professor, can you make the point of the pen a bit smaller? So that way when I go back to watch this video and reread it without you saying it, I can actually understand.
Is it too big here? Too thick?
Yeah, it's too thick. So
overwhelming. Okay, hold on. Let me see if I can do that. Pen property. There. Is that better?
Much better. Thank you.
Okay, yeah. So these as in the APC, these serves as a link between. Anyone want to try to finish my sentence here? Just want to see if you are with me here. We need the APC to link what and what together. Innate and adaptive. Right? Remember, the APC, the macrophage, is part of innate immunity, right? Just eating things up, But then because of its ability to do antigen presentation, it serves as a link, a bridge to the adaptive side, right? To activate the T cells, which ultimately would lead to activation of the B cell, right? So the APC is very important, and I want you to remember that. Okay, towards the end of the lecture, we're gonna come back to this point, like what happens if you don't have any APC, right? How's that gonna cripple your immune system, for example? All right. All right. I hope this whole thing makes sense. I'm just gonna try to zoom out a little bit for you. I don't know if you're able to see it. Yeah, I won't be able to fit the whole thing on the screen, but you know, maybe zoom out just a little bit. And we're gonna go for a break right now. Okay. When we come back, we will take another note. All right. 5 minutes. I'll see you at 9:45. Oh, hello, people on YouTube. If you're watching this right now, you will notice that like it makes no sense because I forgot to press record after our break. But that's okay. That's okay. I will post another video, like an older recordings of the part that was missing. Okay, so you can watch everything there. Okay, but I'm just gonna end the recording here, and you should go check out the second link. All right. If you don't know what I'm talking about, if you're confused, just send me an email and we will sort that out for you.